actually not going to talk too much about the koalas, in fact hardly at all, but just suffice it to say that we are trialling a vaccine in animals that have been relocated due to the building of the Moreton Bay rail link, and we are actually seeing a decreased disease in vaccinated animals. We're now out to the 12 months time point since they got their first vaccination. But anyway, what I'm going to talk to you about today is work that we've done over probably about 10 or 12 years now based around an animal model. But why study chlamydia? Well, basically this here shows what's happened in Australia over the last 10 years, and we've had almost a fourfold increase. And so one of the modelers that I work with has worked out that basically there are 10 new cases of chlamydia in Australia per hour. So by the time I finish talking, there'll probably be 5 new cases. So keep watching the board. This reflects what's happening worldwide. So WHO estimates there's 106 million new infections per year. The infection rates continue to increase despite the availability of antibiotics. So azithromycin and doxycycline are the antibiotics that are most commonly used. And also there's evidence that decreasing use of barrier contraception is contributing to the increased incidence that we see around the world. So the major problem in terms of healthcare costs of chlamydial infections is the inflammation that occurs following the infection. So the actual infection itself is relatively mild. In fact, 75% of infections in females are actually asymptomatic. So you've got a really mild infection in the first instance, but over time, chronic inflammation occurring in any part of the female reproductive tract, which can present as pelvic inflammatory disease, going on to cause blockage of the fallopian tubes, so tubal occlusion leading to infertility, and then quite rarely, but can be quite serious, ectopic pregnancy, where you get implantation of the fertilised ovum, usually in the fallopian tubes, but outside of the uterus. So basically the healthcare costs, the direct costs in Australia are estimated to be about 140 to 150 million. And if you add to that the cost of assisted reproductive therapies, which about 1/3 is believed to be due to chlamydial infections, you're getting between 240 and $300 million health costs due to the effects of chlamydial infections. So just to summarise, these are asymptomatic, so if you don't screen you won't find, particularly in females. And so not many countries around the world actually have active screening programs targeted at the at-risk groups, which are those between the ages of 15 and 29. The healthcare costs are due to the chronic inflammatory damage to the reproductive tissues. So obviously this is the immune system involvement. And so what causes inflammatory tissue damage? We need to know this because we need to make sure that the vaccine doesn't in fact make the damage worse. Current antibiotic therapies have not halted the increase in infections, and studies in British Columbia and Vancouver by Bob Brunham's group have actually shown that targeting at-risk groups with very early antibiotic treatment and surveillance led to a brief decrease in the incidence, but now it's bounced back and they're actually seeing more. So when we write reviews, we all agree with each other that vaccines are probably the best way to go. And so obviously this means activation of the immune system. And so the 2 questions I think we need to answer are, what is a protective immune response that we need a vaccine to induce? And what is the cause of the inflammatory damage which occurs over a period of months to years after what is really a fairly minor and fairly mild primary infection? So we've used a mouse model in our lab, and so mice, when they're treated with progesterone to bring them into diestrus, can be infected with chlamydia. They undergo a standard infection curve, and in BELBCs they will clear around about 30 days post-infection. At this stage, a single infection in mice, and this is one difference to what we see in humans, will cause quite severe pathology. And in the mouse, because the ovary is contiguous with the oviduct, you get this hydrosalpinx occurring, which is something that's very easy to score. So an animal that has hydrosalpinx like this, you would have total blockage of the oviducts and the animal would be 100% infertile. So this is the model that we've used to look at developing vaccines and also to try and understand the mechanisms of pathology. So what have we learned so far from our vaccine studies? Well, this here is a study from quite a number of years ago, and we can see that in this case transcutaneous immunisation, so we've used major outer membrane protein, which is an antigen we use quite often, and in this case the adjuvant was a combination of cholera toxin and CpG, and so you can see the immunised and the non-immunised groups, and obviously we're getting a rapid clearance, so the duration and the magnitude of infection is both reduced by, in this case, transcutaneous immunisation. We also know that a number of different adjuvants can be used to achieve this. So here again we've used our common antigen, the major outer membrane protein, and we've used Iscomatrix, an adjuvant from Vito in Canada, the CTA1DD, which is a modified cholera toxin alpha subunit, inulin from Nick, and compared it to the PBS. So at the moment we're looking at just clearance in vaginal swabs, and you can see that subcutaneous immunisation with MOMP combined with any one of these adjuvants actually reduces the magnitude and the duration of infection to a certain degree. We can use intranasal immunisation, and this is one that I'll come back to. So here again we've used MOMP as our antigen and Iscomatrix, the Vito adjuvant, CTA1DD. And again, delivering the antigen via the intranasal route is able to reduce both the magnitude and duration of infection significantly compared to the PBS controls over here. Something else we've learned is that vaccination can actually prevent ascending infections. So obviously the initial infectious challenge is in the vaginal vault here. The infection ascends and the damage is done up here in the oviducts, which is the equivalent to the fallopian tubes in women. And so here we've got a sublingual immunisation protocol, and over here a transcutaneous, again with CT-CpG, and we see that we can reduce, compared to the other unimmunised groups, the ascending infection up to the oviducts that you see here. We also know that we've got vaccines that can prevent pathology. So again, using our scoring system for mild, moderate and severe hydrosalpinx, in the unimmunised mice after a single infection, usually the diameter of the hydrosalpinx will be 6 or 7 millimetres. Normal is down here, non-infected animals around about 2 to 3. And so these two vaccination protocols, sublingual with MOMP and a modified cholera adjuvant, or intranasal with MOMP plus CpCTG, can reduce to a certain extent the amount of pathology, the development of oviduct occlusion. Now, a PhD student, Connor, in the lab was looking at a number of different vaccine approaches, and what we found was that vaccines could protect against either infection or pathology, and we found this as kind of interesting. But if you look here in red, we've got animals where they have been immunised intranasally with MOMP, CpG, and these animals have almost 80 to 90% infertility, so they've got oviduct occlusion, and yet if you look in the oviducts of these animals, there's virtually no bacteria present. The converse occurs when we've immunised over here with a sublingual approach, and here you've got virtually no pathology, so most of the animals have intact oviducts and would be fertile, and yet over here we've got quite a considerable burden in terms of the amount of IFU of chlamydia in the oviducts. So we've got vaccines which can prevent the infection occurring, we've got vaccines that can prevent the actual damage to the oviducts that can occur. So I guess this raises the question to us is what is the most important if we're trying to go on and develop a human vaccine. So looking at some of the major differences in these two animals, so in the dark grey here we've got intranasal with CT-CpG. These animals were protected against infection, so virtually no bacteria in the oviducts, and yet they developed severe pathology. Here we've got the other group where they were protected against pathology but had quite a high burden of chlamydia in the oviducts. And this is just part of a whole oviduct tissue PCR array, just concentrating on the IL-17 cytokines and their receptors. What we see is in the animals that have very low burden but have developed pathology, we have a lot of IL-17 production, so this is 500-fold above non-infected baseline controls. We also see some increase in some of the other IL-17 members, whereas the animals that are protected against the development of pathology, they have markedly less IL-17, particularly IL-17F, and also a marked reduction in the expression of the receptors. So that kind of indicated to us that the IL-17 family may be involved in the protection against infection and or protection against pathology. Looking at some of these animals in terms of the cytokines that they were making, so here we're looking after immunisation with these various methods down here at cytokine production in the re-stimulated cells from the lymph nodes that drain the reproductive tract. What we can see is that animals that were protected against infection, so virtually no bacteria in the oviducts, but they did develop pathology, they had considerable levels of IL-17 production by cells in the draining lymph nodes, indicating that perhaps IL-17 was required for protection against ascending infection, but could potentially contribute to the development of pathology as well. Now, Chlamydia norodarum can also cause a respiratory infection, so a pneumonia, and if you give Belbsy mice 2000 IFU intranasally, they will lose weight, which will be maximum weight loss about day 10, and then they'll start to recover. So we looked again in the respiratory infection model at a number of vaccine protocols, and you can see that most of these can prevent the weight loss compared to the non-immunised controls. But also we started to see this dichotomy in the lung as well, where we got 3 of these protocols, intranasal, sublingual and transcutaneous, all protected against weight loss, and we know that that correlated with a reduced burden. But there were only 2 of these that actually protected against pathology, and we can score the lung pathology on a scale of 0 to 3, with 3 being the most severe. So you can see that not all vaccines, although they all would prevent the weight loss, were able to prevent the development of pathology, and so sublingual and also the transcutaneous routes of immunisation were quite effective at preventing pathology as well as preventing the weight loss. And when we looked at IL-17 production in these animals, what we found was that protection against pathology actually correlated with a high IL-17 production, so as you can see here and here, and that's the opposite of what we saw in the reproductive tract. And so we were kind of confused about this, but it's subsequently been confirmed that the role of IL-17 in the lung and the reproductive tract does appear to be different, and studies from Tony Darville's lab in North Carolina have come up with the same sort of findings. So what do we know about IL-17 and chlamydia so far? Well, IL-17 is obviously important against extracellular pathogens due to the recruitment of neutrophils. It has been implicated in a number of inflammatory conditions, so EAE and IBD models in the mouse. In terms of chlamydia, there was one study done where IL-17 receptor alpha knockout mice were infected, and there was no difference in the rate of infection. But obviously we need to remember that there are actually 5 IL-17 receptors, so knocking out one may not get rid of all the effects. IL-17 has been shown to be important for resolution of a lung infection. An earlier study that I referred to, this was an antibody neutralization study, and also a number of vaccine studies, particularly from Bob Brunham's lab, have shown that multifunctional cells secreting IL-17 together with either interferon or TNF are important for clearance of infection. So kind of a confusing message perhaps. So we went on to have a look using IL-17 knockout mice, and these are the IL-17A knockouts. What we found is that the actual infection is reduced in IL-17 knockout mice. So here we have non-immunised at this stage knockouts compared to the wild type. We've looked at the area under the curve just to make it easier, and this is recovery of IFU from the lower reproductive tract that you see here. And so the infection in these knockout animals seems to be less in terms of duration and magnitude. When we look at the ascending infections, so now we're looking by PCR at the chlamydial burden in the oviducts here. Again, the ascending infection is markedly reduced in the IL-17 knockouts. And if we look at the development of hydrosalpinx, so the occlusion of the oviducts, again we see markedly less oviduct damage in the knockout mice compared to the wild types after a single infection. So when we came to immunise these animals, we already knew that in terms of naive animals, they develop less hydrosalpinx than the wild types. We can immunise wild type animals and reduce the degree of oviduct damage, but starting from this low base here, immunisation was ineffective in the IL-17 knockouts at further reducing this, the degree of oviduct inflammation. So in terms of the summary of what we found from these mice, we know that the duration of primary infection is reduced. The degree of hydrosalpinx or oviduct occlusion is also reduced. I didn't show you the data, but as you might expect, neutrophil and macrophage infiltration is reduced in the knockouts, and the production of MMPs, which contribute to the inflammation, is also reduced in the knockouts compared to the wild type. And so intranasal and transcutaneous immunisation were able to protect the wild type, but they couldn't further reduce the degree of damage or the magnitude of the infection in the knockouts. So at this stage, we were still not absolutely certain as to what the role of IL-17 was, but obviously it was playing a number of roles in both the development of pathology and protection. One of the things we kind of think is the case is that pathology is driven in naive animals by an overactive innate immune response producing IL-17, and there are a whole list of innate immune cells that can produce IL-17, have been demonstrated to do so. So we've been looking at primary infections and a number of knockouts. So here we have the CD1 knockouts, which lack invariant NKT cells, and you can see that both the incidence of oviduct occlusion and also the magnitude is significantly reduced. And over here we've got the MR1 knockouts, which lack mate cells. There's only 5 animals in this group, unfortunately, because it was just one of those suck-it-and-see experiments. But again, although it's not significant, we seem to have a reduction in the development of oviduct damage. So we're now looking at these cell types in normal animals to see whether they actually do produce IL-17, and this is the subject of some ongoing studies. But to come back to vaccination, we're now looking at what we consider our favourite adjuvant at the moment, and that is Iscomatrix. And when we immunise intranasally with Iscomatrix, and again using the major outer membrane protein as our antigen, we get protection against both infection and the development of pathology. So over here we have mice immunised with Iscomatrix plus MOMP, challenged with chlamydia at 3 different doses. And you can see that the magnitude and the duration of infection is significantly reduced. But over here, if we score the pathology, we're seeing quite significant protection, because I told you that about 2 millimetres is the size of the oviducts in normal, healthy, non-vaccinated and non-challenged mice. So basically this form of immunisation, intranasal with the adjuvant Iscomatrix, gives us protection against the inflammatory damage and also significantly reduces the magnitude and duration of infection. And when we look at cells that we took out of the spleen in this case and re-stimulated them with MOMP, or we can do the same thing with whole-kill chlamydia, what we see is that both CD4 helper cells in the spleen, but interestingly also the cytotoxic or CD8 cells, contain a large number of chlamydia-specific multifunctional cells, which are secreting combinations of IL-17, interferon, and TNF. And this is interesting that this occurs in the immunised animals, but also animals that have recovered from a natural infection, which is the live infection control. So we were quite surprised in a way to see that these CD8 cells were also produced by vaccination, because dogma had always said that the CD4 cells were the most important cell for protection. And as I'll show you in a few minutes, there might even be some data to suggest that these cells are involved in inflammatory damage. So we looked at the previous slide at cells taken from the spleen. So what we decided to do next was to look at the actual cells that were isolated from the reproductive tracts of mice that had been immunised. So what we did here was at day 8 post-infection, when we know the peak of infection has occurred and protection means that the burden is starting to go down. So we digested the tissues, so the oviducts and the uterine horns. So basically all these cells are taken from here up to here. We got rid of the ovaries. And we sorted the cells into macrophages, neutrophils, cytotoxic and helper T cells. So if you just want to concentrate on these two panels over here, what we see is that the cells that are recruited through vaccination into the actual reproductive tracts themselves, both the cytotoxic cells contain multifunctional cells which are producing, in this case by PCR because we couldn't get very many of them, combinations of TNF, interferon gamma and IL-17. In some cases, IL-1 beta was also produced by these cells, which was kind of surprising because that has been implicated in inflammation. Similarly, when we look over here at the CD4 cells, which we've isolated from the actual tissues themselves, we see that we're getting multifunctional CD4s, which are producing interferon gamma and IL-17, and to a lesser extent than the CD8, some are also producing TNF-alpha. So one thing that had always surprised us, and one thing that we had taken advantage of, was that animals that have recovered from a previous infection always have what approaches sterilising immunity if you look at recoverable chlamydia from the vaginal vault. So we use this as a positive control because it's effectively 100% protection against shedding, but we also know that these animals will have severe pathology as a result of that primary infection, so we can use it as a control for the development of pathology as well. And when we looked at animals that had recovered from a natural infection, what we saw was that these animals had a population of cells, some quite significant clusters of CD8 cells, which were CD103 positive as well, identifying them as tissue-resident memory cells. And these were only found in animals that had recovered from a previous infection, and as you can see in non-infected controls, there's absolutely none of these cells present. So again, we were wondering whether these cells were the ones that were important, played a role in protection, or the other option being that these cells played a role in the development of pathology, because we know that these animals, although they are not secreting any chlamydia, already have severe oviduct damage. So we went back to our, compared the animals that had recovered from a previous infection with the animals that have been immunised with the MOMP ISCO matrix vaccine. And what you can see is that in the live animals that had recovered from a prior infection in the grey, at the time of the secondary challenge, they already have these resident CD8 cells in the reproductive tract, and by day 2 after infection, CD4 cells are also rapidly recruited into the reproductive tract tissues. However, if we look at our ISCO matrix and MOMP immunised animals, the recruitment of these cells into the reproductive tract is delayed, in this case till day 8 for the CD4s, and we're starting to see around about day 4 for the CD8 cells. So the cells that are there in the animals that have had a previous infection are sitting there ready to go when you come back with a secondary infection in terms of the CD8 cells, and the CD4 cells are very rapidly recruited, and this recruitment is significantly faster than what we see in the animals that have been immunised with the MOMP and the ISCO matrix. So what we believe now is happening is that in naive animals, potentially in humans as well, that an overexuberant innate IL-17 response is what is contributing to the damage to the oviducts and potentially the fallopian tubes. And we have some evidence that invariant NKT cells and mate cells may be sources of this IL-17, but that needs to be shown. In terms of protection, multifunctional CD4 and CD8 cells, which are secreting combinations of IL-17, interferon gamma and TNF, contribute significantly to protection, and that optimal protection probably requires the early recruitment and retention of immune cells into the reproductive tract so that they're there waiting and ready to go. And so we've got some animals immunised now where we're looking at chemokine treatment and also delivering the final vaccine intravaginally to see if we can bring these cells in and keep them there so that they're in place as you would find in animals that have had a prior infection. So one issue that we still need to resolve was this report by a colleague in Chicago who showed that CD8 cells are actually involved in the development of pathology. So there are a number of potential explanations for this. Ash is always using C57 mice, whereas we use the valve-C exclusively. But he has quite convincing data that CD8 cells that produce TNF are actually the cause of the pathology. So in terms of coming back to our work, what we need to sort out is whether the CD8 cells that are secreting interferon, TNF and IL-17 are actually required for protection, or whether those cells are the ones that are involved in the oviduct damage that's induced by a primary infection, as the studies that have been done in Chicago would suggest. So what are the implications for a human vaccine? What we know from our animal studies is that vaccination can reduce both the magnitude and duration of infection. Vaccination can reduce oviduct damage. And of course, what all vaccinologists do if they don't have funding for a human trial is you get in touch with some mathematical modelers to see if your data actually means anything. So we did some work with David Wilson's group at the University of New South Wales, and basically putting some of our mouse parameters into some models that he came up with, we showed that being able to reduce the magnitude and duration of infection and reducing oviduct damage would have a major effect on the health costs. And this is just one simplified figure from that paper, but if you think here of a critical load needed to infect a non-vaccinated individual compared to a vaccinated individual, and also the duration of infection, you can see that even in the absence of sterilising immunity, if you raise the infectious dose needed to establish an infection and you reduce the duration of infection, then you can reach a stage where the incidence of infection should fall across a population. And I haven't had time to show you, but we know that we can do both of these in both the female model, mouse model that we're using, and also we have a male infectious model, and we know that we can achieve the same things. So I think there is potential to hope that we can develop a human chlamydial vaccine. It doesn't need to induce absolute sterilising immunity to have a major effect on the incidence of infection and also the development of the inflammatory pathology. So I'd just like to thank a number of people. So these are students and collaborators who over the years have done the work, and these are people who have very kindly provided a number of the adjuvants that we've used, and then David is the modeller that we turned to when we didn't have funding for the human vaccine, and also some of the funding sources that I'd like to thank down the bottom. So thank you for the attention. I'm happy to take questions, and I'm sorry I didn't make it yesterday. Nick.
So Ken, thanks for a lovely talk into the complexities of chlamydia. And so I guess the translational question is going to be, given even the differences potentially between the valve-C mice and the black 6 mice, what's the pathway to get this to humans, where people are going to say, 1 in 100 women, maybe they're going to get worse disease, or because of the genetic outbred. So have you thought about using genetically outbred mice or some other outbred model to just prove that you don't see a dichotomy, that we do get all protection?
Yeah, obviously you can do studies in rhesus macaques, but that becomes quite expensive. We are currently doing studies in the guinea pig, because the guinea pig is another model you can use for chlamydia research. It's got a longer reproductive cycle, around about 18 to 20 days, I think, from memory. You don't need hormone priming to get an infection to occur, and you do develop very similar pathology to what you see in humans. One of the other benefits of the guinea pig model is that pathology gets worse with subsequent infection, whereas in a mouse, primary infection gives you 100% at least in the bowel. So we have got studies underway with the same antigen, which we've isolated from Chlamydia caviae, together with the Iscomatrix adjuvant. So that's, I think, will be a step along the way to human trial approval. Certainly there are people, Dorothy Patton's group in Seattle has a primate colony, and for an exorbitant cost she will do studies for you. So, you know, the primate model is there available if you can afford it. I think that if we can show the same thing in the guinea pig, and also in guinea pigs, obviously you can infect and then immunise animals that have had a prior infection. We've done that in the koala, and it doesn't seem to have any effect on making disease worse. So my best bet would be to go to the guinea pig model, and then go and talk to the regulatory bodies.
And you didn't mention that, is there any role for antibodies? I mean, you focused on T cells, so...
Yep. The antibody story is interesting. There's been a lot of work done about 20 years ago where, remember the tumour backpack model, where you can put a hybridoma that's secreting IgA in as a tumour and it's transported into the reproductive tract, or you can inject very high amounts of antibody, and if you've got enough antibody there, you can prevent infection. Rick Morrison's lab has shown that antibody is required for protection against a secondary infection, and it's a very complex model. So I think if antibodies are there at high enough amounts at the time of primary infection, yes, it has a role to play. But once the infection gets intracellular into the cells of the endocervix or in the male, the penile urethra, then you need a cell-mediated response as the major protective effect.
And is the antibody to the MONP as well, or is the primary antibody something else?
Yeah, we've made antibodies against MONP and transferred it, and we get some limited protection. And Harlan Caldwell a long time ago made a number of hybridomas against MONP of different subclasses and showed the same thing. So yes, you can make it against MONP, you can make it against some of the polymeric membrane proteins, the PGPs, and they will also provide protection if the antibody's there in high enough amounts.
So one option would be to try and induce antibody and a T cell response,
yes,
and get synergy.
Yeah. I mean, all these, I didn't show you any of the antibody data, but certainly the Iscomatrix does induce a local antibody response. And of course, in the reproductive tract, the major isotype that's there is IgG, so you're looking at that rather than IgA like most other mucosal surfaces.
Because if this were to go to development, testing antibody would be the simplest way.
Yeah, well, that, I mean, one of the things about vaccine development over 50 or 60 years is that antibodies are always the easy thing to measure. And I mean, even now, people in the HIV field are only starting to measure mucosal antibodies, so going one step further and trying to look at a local CD4 cell response. And I think, you know, we've got some quite convincing data now that those cells we find at day 8 in the reproductive tracts themselves are sitting in the draining lymph nodes after intranasal immunisation, and it's a matter of getting them to the stage quick enough. And it's probably even more important in terms of genital herpes, because a herpes infection, the virus can be in the dorsal root ganglion within 12 to 18 hours. And if you can't get those protective cells into the site, then you're not going to prevent the establishment of infection. So for infectious agents that require a strong cell response, I think you've got to have the cells there at the place where they're going to infect.
Is there not a major pathogenic factor that you would target for the vaccine?
Well, I haven't talked about it because we're trying to sell the rights to these antigens, obviously, but we have a number of other antigens. The reason why we use MONP was that, unlike the Trichomonas cerebrans that infect humans, In the mouse model, there's only one Chlamydia viridinarum, so we can use the MOM. We know we can.